This video is dedicated to SDS PAGE, also called sodium dodecyl sulfate polyacrylamide gel electrophoresis. One of the hazards associated with this experiment is the acrylamide, which is used to make the gels. Acrylamide is a neurotoxin and carcinogen. So in today's experiment, you have to wear gloves and, as always, your safety goggles. And because it's Corona time, here you see I also wear a mask. So to prepare the samples, you'll be working with your samples from the last experiment, the ion exchange chromatography experiment. After you thaw the sample, set out a new set of test tubes, label the test tubes well with the sample names, and into each of these test tubes, you'll be mixing your sample with a loading buffer. This loading buffer contains sodium dodecyl sulfate, SDS, to cover your protein with negative charges, glycerol to make the loading buffer more viscous, and also some bromophenol blue as a blue dye. Next, have a look at how to assemble the electrophoresis apparatus. So here is a cartoon drawing of the mini cell that we're going to use. There is a large buffer chamber, like a large box, and into that large box you'll place the so-called buffer core. The buffer core is colored white and has a wire. And in order to position the gel, if you only have one gel, there's also a buffer dam and a gel tension wedge, and finally the mini cell lid with the electrodes. So in the lab, what you see here, I'm handling the inner buffer chamber with the electrodes, and I have that bigger chamber, and there's only one way to position the inner buffer core into the larger electrophoresis box. Next, I'm cutting open the pouch to get to the gel. The polyacrylamide gel is embedded in a plastic cassette. That plastic cassette has a tape on the bottom, so this white tape, it has to be removed because the white tape covers a strip of polyacrylamide gel. And then you also have to remove the comb. The comb makes sure that you have individual sample wells, and then carefully place the comb back into the pouch because all the plastic pieces and the pouch itself needs to be discarded in the special waste container for anything that came into contact with the polyacrylamide gels. In this setup, I'm using a second gel. So again, you see how I open the pouch with the scissor. Then as I remove the gel, don't forget to also peel off the white tape. And then I also have to remove the comb on top. Here is the wedge. The wedge has that white colored handle, and I need to tilt the handle to get the wedge into the electrophoresis box, and to secure everything, firmly pull the wedge towards the front. If your apparatus is put together correctly, everything is held in place very firmly. If you're working with only one gel, then you need to use that plastic dam, that extra plastic piece. It has the label "Do not discard." It serves as a placeholder for the second gel. Here you see how I'm pouring buffer into that inner chamber, into that inner buffer core, and I'm pouring all the way to the top to cover the wells of your gel. So I'm pouring above the white bridge so that the inner buffer core, that inner chamber, is filled all the way to the top. Then I'm checking that there is no leakage, that the inner core is really separate from the outer, and only then am I pouring more buffer, the same running buffer, into the outer chamber. I can pour from the front, it's connected to the back, so don't be alarmed if the buffer that you pour in the front into the big chamber ends up all the way distributed in your electrophoresis chamber. I don't have to pour all the way on top for the outer buffer. Then to test the whole apparatus, I'm connecting with the lid. There's only one orientation, and the color coordination also helps to make the proper connection. And this is just a test run to see if everything was set up properly. And I'm looking for how the voltage increases slowly but steadily. And then I'm checking for bubbles. So you can see there is a wire at the bottom of that buffer core, and that wire is the anode of your electrophoresis chamber. And when bubbles evolve, then your electrophoresis setup was done properly. Here you see how I'm rinsing the gel wells. So the loading wells where the sample is going to be with some of the buffer. And then I'm getting ready to actually load the real sample. You can use a P100 or a P200 pipette, or if you're loading only 10 or 20 microliter, a P20 pipette will also work. The first sample that I'm loading actually is the standard. I'm using my finger to press the tip towards the front of the gel and to also be steady and to guide the pipette tip into the first well. Always take a new pipette tip. The standard, you only need about 10 microliter. For the sample, you can use more, 10, 20, 30 or 40 microliter. Before I'm actually applying the sample, I'm mixing the sample really well because it's the sample and the 2x loading buffer. And then again, you can see how I'm using my hand, which guides the tip more firmly into the well to have more control. The pipetting is really slow but steady because you don't want your sample to spill out of the well. So next you see a side view. I'm done with about half of my samples. So mixing the sample with the loading buffer really well is important. So pipetting up and down a few times can help with that. And then again, you see how my fingers are helping to guide the plastic tip into the well, and how slow and steady pipetting without blowing out your sample at the end can help to fill the loading well in a steady manner where you don't have any spill, because each sample belongs in its very own well. It is very important to keep track of what you loaded into the well. So here is a loading scheme. If you used a different loading scheme, simply write it down in your lab notebook. The next step is the actual electrophoresis. So in order to set the parameters, you click on the Mode button. The Mode button cycles you through the key parameters, the voltage, the time, and the current in ampere. To change the setting after you clicked on the Mode button, just press the up and down buttons. Here you see a typical setting, 225 volt, and a time of about 35 minutes. You can also set 30 minutes. You don't have to set the amperes, but it can help to set a really high value. And then finally, click on the constant button to toggle between constant amp and constant volt. These pictures show you snapshots on how the gel evolves at different time points. Then finally we have to crack open our cassette. So the whole apparatus was assembled, and here you see how I'm using that thick spatula and I'm going around the rim between the two halves of the cassette, and with every motion you will hear a loud cracking sound because there are notches between the two pieces or between the two halves of your cassette that are broken open. I already prepared a tray with the buffer, the 100 millimolar sodium phosphate buffer at pH 6.75, and I'm using the thick spatula here to press down in that area that was covered by the white tape. So there is an area where you can push through in order to deposit your gel into the tray. And so far this tray just contains the buffer. To stain for PPO activity, you need more than just the buffer. So here is the gel, and we only see the standards so far. It's a pre-stained standard. But then we add dopamine, enough dopamine so that the final concentration is 10 millimolar. And here you see snapshots after different time points, and also a snapshot after adding the second staining reagent. You can take a photo with your smartphone, but also in addition, we have that imaging device with which you can also take photos, which also has some software to further analyze the gels. But the gel analysis will be covered in another video. So have a good day, and I'll see you next time for a video Zoom session or an actual on-ground lab. Bye-bye.